Welcome everyone to this webinar of today, the RNA stability webinar. So my name is Mireia Alegra and I'm the marketing manager at the Isogen Life Science. And I will be hosting this event today in collaboration with Inactive Blue. And this webinar of RNA stability is the first webinar from a series that we are doing, that it's the NGS series. So you will have from now until June different webinars coming based on the different workflow of NGS. So we are starting with RNA stability, but then we will move to DNA RNA extraction, sequencing, and then we will do RNA sequencing user day at the office on the 10th of June. If you want to know more about it, the invitation, like how to register and stuff, you can always get in touch via support@isogenlifescience.com. And also I will be doing some marketing, so you can always get invitation from our side. But first of all, I will briefly introduce Isogen Life Science for those that don't know about us. We are a distributor company founded in 1987, so a long time ago. We are a solution-driven company that provides equipment, reagents, consumables, but also services in the life science field, focusing mostly in genomics and transcriptomics, but also on cell biology, proteomics, and molecular diagnostics. With that, we have like a whole workflow of solutions from sample to results. So we have from cell culture, cell lines, cell counting, imaging systems, to more like DNA, RNA, stabilization, and extraction kits, and the separation and detection techniques that go along with this. So we have PCR thermocycles, next generation PCR, digital droplet PCR, microvolume spectrophotometers for DNA quantification, but also NGS solutions, Sanger sequencing, etc., and also sequencers. We have along all this workflow, the consumables needed for this, and services as well for NGS RNA sequencing services. But for the topic of today, we will be focusing on the RNA stabilization concept. And for that, we have our guest speaker, Jovan de Somple. He's the co-founder at Inactive Blue, and we are the distributor of Inactive Blue in our market regions. And he will be focusing on the RNA challenges and the technologies that have been developed for this as a solution, for example, the RNAwait. So for that, I would love to give the floor to Jovan de Somple. So like now, if you can start sharing your screen, we can continue with the real content of the webinar. So I will stop sharing. Yeah, perfect.
Thank you very much for having us. 1 second. All right. I hope my screen is clear.
Yes, it's visible. Yeah, thank you.
Okay, today I'm going to focus mainly on our latest product, RNAwait, as a solution for preserving RNA in tissues. So the problem is that as soon as a tissue is removed from the body, that RNA degrades rapidly. This is because RNases gets activated and they start to hydrolyze the RNA. At the same time, cells alter their gene expression due to changing environment. The temperature changes, oxygen levels changes, etc. So both cellular stress and nucleases result in the fact that the RNA profile that you get is not representative of the in vivo situation. So this leads to poor RNA integrity and inaccurate molecular analysis. And this is really a big problem if you want to study in vivo situation of RNA. So there have been solutions in the past developed, and the historic solution is formalin fixation and paraffin embedding. This is really the gold standard for pathology and immunohistochemistry. However, this creates severe problems because it fragments RNA and it limits molecular studies. So while it's perfect for morphology, it is not a good solution to do downstream RNA analysis like RNA sequencing, quantitative PCR, or digital PCR. An alternative solution is snap freezing of the tissue in liquid nitrogen. This really preserves RNA well if done immediately, so that's important, but it creates the problem, a logistic problem of access to freezing infrastructure, which is not always possible on site. Or if you do in-field studies, of course, this is not possible. But perhaps even the biggest problem of relying on freezing is that as soon as you thaw the tissue sample, RNA is rapidly degraded because RNases are activated and no longer kept under control. So while it preserves RNA in perfect quality, it poses logistic issues and really creates a operational workflow if you want to process the frozen sample downstream. So better solutions must be made available, and we have developed one. So RNAwait is our solution to solve this logistic bottleneck. It's really a simple solution that as soon as you embed a tissue in the buffer, it quickly permeates and it stabilizes RNA. So there's no immediate need to process or freeze the samples. So you can extract the RNA at a later time point. It's a very flexible and easy workflow. First, you cut the tissue into pieces that are maximum 5 millimeters of width, so half a centimeter. And this is very important to make sure that the preserving buffer can penetrate the tissue deeply, quickly, and effectively. Half a centimeter is our recommended width of a tissue. However, for some tissues that are difficult to penetrate or that are rich in nucleases, we even recommend thinner sections of 2 millimeters. Then you take your sectioned tissue part, typically a cube, and you incubate it 8 to 24 hours in the fridge in the buffer. Typically, this is done overnight. It's not very important how long it is, just sufficiently long so the tissue is fully impregnated by the buffer. A third step, and this is an important one, where some people make a mistake that they then freeze the buffer with the tissue. And in contrast, they should remove the tissue from the buffer. And from that moment onwards, it's totally preserved. So you can keep it at room temperature. You can freeze it. You can put it in the fridge for different durations, which I will explain later in this webinar. But so you have to take it out from the buffer and then do whatever you want. Put it in a new container or freeze it under the likings of your preference. And then the extraction is really up to you. If you freeze it, you really have indefinite time to do that extraction. If it's in the fridge, you have up to 30 days before you engage in that important extraction procedure. This figure shows the exquisite performance of RNAwait, and you see that the same tissue, namely murine heart and murine kidney, have been extracted fresh, so that's blue, or have been treated with RNAwait and then stored in the fridge for 1 month at elevated room temperature, 25 degrees for 1 week, or at elevated temperature of 37 degrees for 1 day. And here we measured using RT-qPCR 2 genes, NAPC5 and CD81 in both tissues, and you see that CQ values on the y-axis are almost identical, irrespective of the processing and the storage of the tissue. So whether you extract immediately from a fresh tissue or you preserve it first in RNAwait and then you store it at the durations mentioned in the legend, you get the identical results. And this is, of course, what you need, that there are no changes and that the RNA is not degraded. So it's a very powerful, but at the same time, very useful and user-friendly method to preserve RNA in tissue. What is also important is that once you have preserved the tissue in RNAwait, that it withstands at least 5 freeze-thaw cycles. I think this is important because that means that the same tissue, you can cut a piece and then process it, freeze it back, thaw it again, take another piece. So especially for biobanking projects, or those of you that are interested in biobanking, it means you don't need to proportionate or to aliquot upfront, and that freeze thawing not at all further degrades or degrades at all the RNA in the tissue. Importantly, again, you first have to, of course, to overnight incubate in a solution, and then you can do the freeze thaw cycling up to your liking or when it's operationally needed. What is also possible after you have impregnated the tissue in RNAwait is that you still can do paraffin embedding and formalin fixation and do H&E staining. So the tissue morphology is largely preserved once a tissue has been preserved in RNAwait. Importantly, this is only true for H&E, as RNAwait the nature's proteins, this tissue is no longer suitable for most of the immunohistochemistry based on antibody-based detection. So this solution enables not only molecular analysis, so high preservation of RNA, but also preserves tissue morphology if you're interested in this for your application. So a question I often get is how much RNA can reliably extracted from an RNA-weighted treated tissue? And then the answer is really depends on the tissue type. You have tissue types like liver, kidney that are highly metabolic and you really get a lot of RNA. And there are other tissue types like adipose or fat tissue, cartilage, bone that are very poor in RNA content. But just to give you an overall idea, if you cut your tissue into pieces of about 0.5 centimeter in dimensions, so that's the representation on the left, that's around 100 milligrams of tissue, and you typically can extract 10 to 100 micrograms of RNA. And that's really a lot. If you know that most molecular analysis adopt an amplification strategy, this is way too much for a regular analysis. That's why some of our customers preserve very small tissue fragments, like 1 by 1 by 1 millimeter. That's about a milligram of tissue. And you can still reliably and with high quality extract RNA for 100 up to 1,000 nanograms. And this is still largely sufficient for an experiment to do RNA sequencing, reverse transcription quantitative PCR, or reverse transcription digital PCR. Another question is that we sometimes get is depending on the RNA extraction procedure that the concentration is too low. Maybe the yield is fine, but the concentration is too low to start a molecular process. Well, also there, several options exist to increase the concentration. You have the classic alcohol salt precipitation of the RNA and resuspension in a smaller volume, so that creates a physical concentration effect. But you can also use very handy ultrafiltration columns where you remove the buffer and then you turn the column and you get a concentrated version of the RNA. Or some other users have adopted a vacuum centrifuge with modest heat. For instance, you apply heat and a vacuum to 37, 40 degrees, and quickly the water is evaporated and you get a concentrated version of high quality RNA. Importantly, all these three methods don't jeopardize the integrity of the RNA if you do it well. So low concentration should not pose a problem for any downstream molecular analysis. Another very important question is how to evaluate RNA quality. Well, if we discuss RNA quality, it's important to make a distinction between integrity, so if the RNA is physically intact, versus purity. Are there any inhibitors or impurities still present in your sample? The good news is that if you use a RNA extraction kit, that typically most of the impurities are removed, so you should not worry about them. But there are molecular tests to detect whether inhibition is still present. Typically, this is done by spiking in a exogenous RNA molecule and observing that the CQ value in a qPCR, for instance, is exactly the same as you would spike it in water. If that's the case, there are no impurities or inhibitors present. Some people rely on a spectrophotometer and they calculate the so-called 260-80 or 260-30 ratios that are at best indicative of protein or DNA contamination. Please beware, we don't recommend using these ratios because one, they are very insensitive to impurities, and secondly, these ratios are really depending on the pH of your solvent. It's known that water becomes acidic over time, so these ratios do change and are in fact not reliable. So we don't trust these values, we never use them. When we discuss integrity, so physical intactness of the RNA molecules, many different strategies exist, and the two most important ones are looking at the integrity of the ribosomal RNA molecules, 18S and 28S. For instance, using electrophoresis or capillary electrophoresis, you have instruments like tape station, fragment analyzer, the bioanalyzer, etc., where a quality score like a RNA integrity number, RIN, or RNA quality index, RQI, is calculated. This is looking at the ribosomal RNA, and the assumption is that the quality of the ribosomal RNA represents the quality of the messenger RNA, which is typically the case, but not necessarily. That's why an alternative method that was developed is a qPCR-based test, where you measure the concentration of the 5' beginning of a messenger RNA and the 3' end, and if the RNA is intact, and you adopt oligo-DT-based reverse transcription cDNA synthesis, you will get to the same concentration of the beginning and the end of that transcript. We and others have published that. If you're interested in learning more methods or the relative merits of one method over the other, please have a look at our paper that was published a while ago in Nucleic Acid Research, where we not only present methods to determine the quality of RNA, but also indicate the impact RNA quality may have on your results. Discussing RNA quality and RNA integrity then immediately comes the question, what is acceptable RNA integrity? And I really want to warn you, don't get fooled by the RNA myth. Maybe you have a colleague or a core facility manager that says, yeah, we need a RIN of 8, or we need a RIN of 9 or 6. Some even say 5 is good enough. And really, there is no clear answer because it depends on so much factors what acceptable RNA quality is. Of course, we all want ideal quality, and this is exactly the reason why we developed this product. But what is more important is that your samples are of similar quality. It does not need to be perfect because the level of degradation or RNA quality that impact your results really depends on the expression difference of the target you're studying, the so-called effect size, the within group expression variability of your target. It's fair to understand that if it's very robustly regulated, that you can see smaller differences, while there is already a lot of variability at baseline, it becomes more challenging to find a gene as a biomarker. It also depends on the target abundance. If you have a lot of RNA versus little, then a little bit of degradation can immediately destroy the target and make it non-detectable. It also depends on the target sensitivity to degradation. Many different genes have different sensitivities with respect to degradation. Of course, it also depends on the RNA quantification method. Long read RNA sequencing, and that may come back into one of the next seminar series by Isogen, of course, requires higher quality physical integrity of the RNA than, for instance, a qPCR assay with a very small amplicon. It also depends on the number of targets in a biomarker test. If you're looking at more targets, you're more robust against fluctuations, etc., etc. So it really depends. Don't get fooled by an answer. You have to test for yourself what is the lowest level of RNA quality and still call a positive sample positive and a negative sample negative. If you want to know more about our product and how we appreciate RNA quality, please have a look at the instructions for use, the manual of RNAwait. You can find it on our website if you scan the QR code, or if you simply go to inactiveblue.com. And we have a beautifully laid out brochure with the performance and the other metrics of our product. To finish off, I want to indicate that we don't only have a solution to stabilize and protect RNA in tissue, we also have a dedicated solution for blood. If you're interested in extracting cellular RNA from whole blood, so that's not plasma or serum, but really whole blood, so RNA from blood cells in the blood. We have a solution for venous blood with a vacuum tube on the left, or capillary blood, a small microtube where you use a lancet to draw the blood from the fingertip or the heel. And this solution stabilizes RNA for up to 30 days in the fridge or 3 days at elevated room temperature of 25 degrees. And again, we're proud of that. It withstands up to 5 freeze-thaw cycles without any impact on RNA quality. And then finally, our DNA RNA defend buffer, which is an all-purpose buffer, mainly suitable for other fluids like saliva, urine, swab, but it can also handle more complex samples like stool, soil, etc. It's perhaps the most powerful buffer that we have, and it allows perfect RNA integrity up to 30 days at 37 degrees. So this is really preserving RNA and complex tissues very well. With that, I want to indicate two additional sources of information, our website, or of course, if you have any questions, please speak to an Isogen representative. We're happy to work together, and I'm happy to answer any questions you may have today. Thank you for your attention.
Thank you very much. It was super clear. Is there any question in the audience, maybe like via chat, or you can turn on your microphone also as well? Not for now. Well, I have one question. So you talked about RNA preservation. Is DNA and proteins also preserved during this process?
Yes, it is. DNA and proteins are preserved, but as I mentioned, the proteins get denatured. So it's still useful for protein work like Western blot or mass spectrometry or 2D separation. But for methods that require an intact antigen, like antibody-based detection, RNAwait is not the solution you may want to use.
Okay. And is it compatible to all RNA extraction kits, or which ones are compatible with?
In principle, it's compatible with any RNA extraction method. Of course, we have not tested all methods, so we do recommend the user to first test on a non-precious sample whether high-quality RNA can be extracted, but in principle, it's compatible. And the reason is that once the tissue is preserved in the buffer, you take it out, and then the buffer no longer plays a role. It's simply an impregnated tissue that is perfectly suitable for downstream extraction.
Okay, clear. Thank you. Okay, we see some thanks in the chat, but not real questions. Well, I think it was very clear from your presentation, so that's also really good to mention. If there's any question in the meantime or after this presentation, please, as he said, you can always get in touch with us or with them, and we will take care of that. So yeah, maybe we can stop it here, and thank you all. Well, there's a question very fast. Nathalie, so is there a need to wash the tissue before storage?
No, and in fact, even we recommend against it because the RNAwait is a high salt concentration buffer, and it's a reversible RNA preservation method. So if you would take the tissue out of the buffer after overnight impregnation, and you'd put it into water, the salt would get out of the tissue and you would reactivate the RNase. So please don't wash the tissue. At best, we recommend you take a dry tissue or cloth and you remove the remaining buffer that is there. It's not strictly required, but if for some reason excess RNAwait buffer is problematic or you don't want it, just dip it off with a tissue and that's it. No washing. Very good question. Thank you.
Thank you, Nathalie. Okay. Any more questions?
Maybe another remark from the Lexogen side. That's what we also hear. We really value highly what you and Dennis, whole Inactive Blue team has developed. Of course, we didn't test extensively everything, but being the next step after the RNA preservation, it's crucial for companies like us, whether you sell our products or we provide services to customer, that the RNA integrity is preserved. So the consequence otherwise can be terrible. Even some people publish papers and we actually question sometimes some data there, and some causes can be different, but RNA preservation is key. And it looks like a very small product or high salt solution, but it's very important. And all the tests that Jo has performed, the publications he mentioned are very, very important. And it's very important when you have a call with your customers to highlight this before even the experiment starts. So really, thanks for the presentation. It was also very, very clear and easy to follow. Thanks, Jo.
Thanks, Filippo. Great job.
Thanks, Filippo. Okay, then maybe we should just stop it here. Thank you for the questions and thank you for the discussion. If there's anything, as I said, don't hesitate to contact us, and we'll see you in the next webinar. You're more than invited. Thank you very much to everyone. Have a nice day.
Bye.